start with, if it works, yeah, with a brief history of the company. So Genomic Vision was founded in 2004, so we're pretty old for a young biotech. It was originally created as a spin-off from the Pasteur Institute in Paris. I'm sure most of you are familiar with this institution. So Aaron Bensimon, our founder, used to be the head of the genomic instability unit at Pasteur. That's where he discovered our proprietary technology, which is called molecular combing of DNA, which I will explain later. So the company was originally created as a spin-off from the Institut Pasteur in 2004. And for the first 10 years of our existence, we've been in stealth mode because we were only an R&D company for the first 10 years. And we recruited our commercial operations team only in 2014. So for the first 10 years of our existence, we've been solely focused on developing the technology that had been discovered at Pasteur and transforming it into a commercial product. In 2006, we signed an agreement with Hôpitaux de Marseille for our first test, which is a test for FSHD, which is facioscapulohumeral dystrophy. It's a rare orphan disease. And then in 2010, we signed a transformative deal with Quest Diagnostics, which is the world leader in diagnostics. They are our main shareholder and our main partner in the US, because we don't commercialize our tests directly. We design them, we develop them, we manufacture them, but we don't take care of the direct commercialization to patients. We only commercialize to academic institutions, but I will come back to that later. Then in 2014, we IPO'd on the French stock market. We are a listed company. We had to raise capital to finance our transformation into a fully commercial company, and that's the main reason why we had to IPO. And you can't see it here, but the main news for this year is that last month we signed a technological collaboration with AstraZeneca, which we are pretty proud of. They will use our technology in their oncology effort, because our technology can be used as a screening tool for oncology drug candidates, but I will come back to that later. So, molecular combing, this is our proprietary technology. That's the reason for the creation of Genomic Vision. It was originally discovered by Aaron Bensimon at Pasteur, as I said. What I like about this technology is that it's pretty easy to understand, pretty straightforward. So basically what you can see on the left-hand side of this slide is that we start with peripheral blood most of the time. We extract B cells and T cells. We need roughly 1 million cells to start with. Then we put these cells in agarose plug, and then we digest enzymatically the cell wall and all the organelles and everything. Then we're left with pure DNA and agarose plug. Then we digest the plug and we're left with pure DNA in solution. That's the first part, that's the extraction process. Then we put the DNA in a small reservoir that you can see. It's the second step of the process. And then we dip a silanized cover slip in the reservoir, and thanks to the surface tension, the double-stranded DNA nicely attaches by one end to the cover slip. So then when you very slowly withdraw the cover slip from the reservoir, thanks to the surface tension at the meniscus, it nicely stretches and aligns very long DNA fibers, and you get roughly 300 copies of a full human genome on one single cover slip, which is pretty good in terms of coverage. So you can see the third step is a nice illustration. The metaphor I like to use is that it's like when you're swimming underwater, then when you reach the surface, it nicely combs your hair backwards. Thus the name Aaron shows for the technology, because it's like combing, because it stretches and aligns the DNA fibers. And we get much longer reads than with competing technologies like next generation sequencing, where they have very short reads and then they need to stitch the pieces back together. We can have fibers as long as 10 megabases, which is several orders of magnitude longer than what you get with other technologies. Once you have this nice cover slip with roughly 300 genomes on it, then we have our own proprietary scanner with which we scan the cover slip, that I will explain that later. So you have here the steps. So as I showed on the previous slide, we have the extraction and the combing, which gives us a cover slip with the DNA fibers. Then it's kind of like fish. We hybridize the DNA fibers with fluorescent probes. that we design, and that's, I like to use the metaphor of the Lego brick. We design a Lego brick that attaches to a specific sequence we want to identify in the genome, and it's fluorescent, so if it attaches, then when we illuminate the slide in our scanner, we can take very high resolution pictures of the cover slip, and then the last step after the scanner, the Fiber Vision scanner, we have our own proprietary software which is called Fiber Studio that automatically detects specific patterns for which we are looking. And these patterns are biomarkers of diseases we are looking for. Our first test, as I said earlier, the one we partnered to Hôpitaux de Marseille, is a test for FSHD, but then we developed other tests for rare diseases such as SMA. We have a carrier screening tool for SMA, spinal muscular atrophy. And we also have 3 tests in oncology that are already commercial stage. 1 test for breast cancer, 1 test for colon cancer, and 1 test for cervical cancer caused by HPV infection, for which we have an ongoing clinical trial going in the Czech Republic. So, the 2 pillars of Genomic Vision. As I briefly mentioned earlier, our business has 2 legs, which is pretty convenient. The first one is IVD, as I mentioned. So for facioscapulohumeral dystrophy, breast cancer, which was our first test that we partnered to Quest Diagnostics in the US, SMA, and human papillomavirus. But since last year, we added another pillar, which we call LSRT, so Life Science Research Tools. And that's the part I briefly mentioned earlier with AstraZeneca, because we also sell our technology, not tests, our platform, to pharmaceutical companies that use our technology to have a real-time assessment of replication events. So it's very convenient for pharmaceutical companies that work on replication inhibitors and DNA damage and repair products such as PARP inhibitors, for instance. And that's the reason why we signed this technological agreement with AstraZeneca. So in this second part, as I said, DNA replication analysis, but we also work actually already with the gene editing and gene therapy companies, because our technology can also be used as a quality control tool for gene editing companies and gene therapy companies, because as we can detect specific sequences in the genome, it's very convenient for them, because right now what they have to do is that each time they make an edit, either with CRISPR or TALEN or zinc finger or even with a viral vector, they have to resequence everything with NGS, whereas with our technology, as it's a direct optical visualization of DNA, it's much easier, much more robust. And also, I think one of the key points is that we don't rely on PCR amplification. So for very rare events, there is no bias involved because we don't amplify. What you see is what you get. It's, again, direct optical visualization. Here you can see a nice example of what we get after the process is complete. So again, extraction, then the combing, then the hybridization, then we put it in our proprietary scanner, which is nothing more than a very high-tech microscope. And then we get these very nice pictures with colored probes. And that's what we call the genomic Morse code, because it resembles a Morse code or even a barcode if you wish. And then the software is able to recognize these specific patterns, which are biomarkers of genetic disorders. In this case, you can see a very complex sequence that is several kilobases long. Our minimum resolution today is 1 kilobase pairs. So we actually don't compete with NGS for SNPs, single nucleotide polyformisms, because we can't see them. We cannot see anything shorter than 1 kilobase pairs, sorry. But we can go up to several kilobase pairs. So basically, we bridge the gap between NGS, which can see SNPs, and normal fish that can see chromosomal aberrations, but which is not very good for highly repetitive sequences. So the GMC, as I said, again, you have to think of it as a Lego brick. So we assemble a sequence that is complementary to the DNA sequence we want to look at, and that will nicely attach to the specific sequence we want to look at. we have a service called GMC on demand, for instance, for gene therapies. As they know what their payload is, we just have to design a complementary sequence to the sequence they want to insert. And once the editing process has been complete, we can very easily extract cells from the patient and then see if this GMC attaches to the sequence. And if it does, and if we can see it on the picture, it means that the editing process has been properly completed. And we can also, in the process, check that there hasn't been any off-target events, which is the main concern. You might have read last week a Nature article on CRISPR, which sent CRISPR therapeutic stock down by a few percent because they questioned not the efficacy of CRISPR, because everybody is convinced of its efficacy, but people start raising questions about potential off-target events. That's something we can look at, and we are actually building a relationship with one of the 3 top players in the field that we will announce pretty soon. This is a completely different application. I won't take too much time on this slide because it's a bit, it's not very clear in my opinion, but this is another application. This is for the replication study. So that's something we do with AstraZeneca. Basically, our technology can provide a direct visualization of DNA. Here, what it shows is an origin of replication. And then we can simply look at the speed at which the replication fork advances. So it's very useful for pharma companies, especially the ones that want to inhibit replication, because they can simply take a cell culture, for instance, apply the drug, and then we look at the replication events and we see if it had an impact, meaning we can see whether the drug actually slows down the replication fork or not, which is very useful. This is another application I mentioned earlier for gene editing. In this case, this one is much more self-explanatory. Here we wanted to look at a deletion with CRISPR. So as you can see at the bottom, on the left, you have the picture before the deletion and you have the green dot in the middle, which is roughly 1 kb. I think it was a little more than 1 kb sequence that they wanted to cut using CRISPR-Cas9. And on the right, you can see that the green dot in the middle disappeared, which means that the sequence they wanted to erase, to cut, was properly erased. So as I said earlier, it's a very easy, straightforward, visual way to check that gene editing processes have been properly completed. This one is very interesting as well. It's another application. So, as I said earlier, we have an ongoing clinical trial on human papillomavirus in the Czech Republic with roughly 200 patients, for which we've had very encouraging preliminary results that we presented during our R&D day 2 weeks ago. So basically here the goal was to visualize the integration of the HPV DNA within the host DNA, because what we want to prove is that we want to correlate the number of integrations of HPV's DNA and the severity of cervical cancer, because there are tons of literature that correlate HPV and cervical cancer, but we wanted to find a biomarker. So on the left in the picture, you can visualize, so the HPV's DNA is the small portion, the blue, red, blue code, and in green, it's a simple DNA fiber that we've counter-stained in green to have more contrast. And on the second picture, you can see that there has been several integrations in the DNA. And the other thing we do is that we look at where it integrates, because there is a high probability that HPV integrates in fragile sites, which makes it even more oncogene. So that's another application that we are working on. Basically, our goal is to transform it into a routine test. We don't want, obviously, to directly compete with Roche Diagnostics or Hologic, because they already have the market. We want to be kind of a reflex test that they can do in addition to the test they are already conducting, just as again as a marker of the potential severity of cervical cancer. I wanted to spend some time on Quest because, as I said earlier, I mean, our partnership with Quest has really been transformative because until 2010, when we signed the deal with Quest, we were solely an R&D company. And the fact that we managed to sign the deal with them kind of validates the intrinsic interest of our technology, first. Second, it gave us access to the US market because they already commercialize our test in the US, because we don't directly interact with patients. We let Quest do that in the US. Only in Europe, we interact with patients in Marseille for FSHD and in Leiden University in the Netherlands for FSHD as well. But for the rest, we let Quest take care of that because they have a much broader reach than we do. So there are three pillars. First, they gave us R&D funding, which is a good thing because at that time we needed some more capital to conduct our R&D effort. As I said, there was a commercial license involved. They have an option on all the tests we develop in the US to commercialize them. And the third pillar was the equity investment. They are currently our majority shareholder, and they have a board seat, so we have a very tight and robust relationship with them. I like this slide. Actually, I should have started with this one because we're pretty proud of this. So over the last 10 years, we've been able to secure pretty nice partnerships with very well-respected institutions. As you can see there, we have a partnership with Harvard Medical School. We have a partnership with the Broad Institute as well, that is not here. We work with the MIT, and also we have a partnership with MD Anderson in cancer. And in Europe, we have a partnership with the Imagine Institute, which is the biggest rare genetic disorders research cluster in Europe. So we are pretty proud that our marketing and sales team and our BD team were able to secure such partnerships because first, they generate sales because we sell them our platform, actually our two platforms, the combing platform and also the scanner, which is nice. And second, it gives us visibility for the pharmaceutical companies because then the idea is to write joint articles with them that gives us materials to prove all the applications in which we want to develop the technology. So this is for the first part, but I just wanted to take some time to give you a more detailed view of our offering because until now I focused on the technology, but I want to show that now we are not only an R&D company, we actually have commercial products. So here on the left, you can see the combing platform. It's not very complex, this one. It's only the equipment that does the combing part. So it dips the cover slip in the reservoir and then it withdraws it very slowly, as I showed earlier, to do the combing. And behind you have the Fiber Vision platform, which is the scanner, which takes very high resolution pictures of the cover slips, as I said earlier, and that gives us the pictures that then we can analyze with our software. And at the bottom, you have the disposables. So we work on a razor blade model, kind of, or Xerox model. Basically, we sell only one piece of equipment, but then we secure revenues through the sale of reagents and consumables. This one, I don't want to talk about it. This one is interesting. It's very early stage. This one, we would like to prove, for instance, with the exposure to UV light, we want to look at the impact of UV on DNA and where it breaks DNA and the repair process that happens afterwards. This one is important, and that's actually the one I wanted to finish with. So this is our service offering, because one thing we realized is that it's very hard to sell pieces of equipment, heavy equipment, to pharmaceutical companies and to academic institutions, because our platform costs roughly $300,000 a piece. So it's not always possible for small core facilities to have them buy our technology. So what we decided to do is to also offer our technology as a service, which is a path that many companies follow nowadays. So we have several offerings. So at the top, you can see the workflow I showed at the beginning. So from extraction to Excel spreadsheets with all the results. And we let the customer several possibilities. They can have the combing platform and then send us the cover slips, which is much easier than sending blood, because once the DNA is fixed on the cover slip, it's much easier to carry around. And then we take care of the image acquisition, and then we give them access to our software on a software as a service basis, which allows them to conduct the analysis on premises. Or we can take care of the extraction, the combing, and the image acquisition and let them do the analysis. And the third service is a sample to spreadsheet service in which they basically send us blood vials, and around 1 week later, we're able to send them a complete report with all the elements they are looking for. So that concludes the first part of my presentation. And now I will turn it to you. I wanted to make it as interactive as possible, so if you have questions, feel free to ask them now. With Curie, it's a very early stage collaboration now. Actually, it dates back a while. Honestly, it's not very active at the moment. Unfortunately, I'm not the one managing this account with Curie. Aaron knows much better than me. I know that it's been very active 1 or 2 years ago, but at that time I hadn't joined the company yet, but I would be very happy to send you a detailed answer on that. First, the idea was to sign, as I showed in my slides, I need to find it again. The first step was to sign as many collaborations as possible, because you know how it works. I mean, you need research, you need publications, you need articles to validate your technology. So when the company was created, we had the chance that Aaron was pretty well known, at least in Europe, as the head of genomics at Pasteur. So it helped a lot, at least in Europe. Then in the US, we have a team of application scientists and marketing and sales people who spend their time traveling around the world and visiting these institutions, and it takes a lot of time. Each time we want to sign a new partnership with an academic institution, it can take up to 2 years. We have to have several meetings with them, usually. Then we give them a proof of concept, free of charge. So they send us a few samples, we conduct an analysis, and then based on these results, they decided that if they want to collaborate. And only at that stage, we can start discussing about the possibility for them to buy a platform. So the lead time is very long, but on the other hand, this step is absolutely vital, because unless we have academics working with our technology, it's very hard then to convince pharmaceutical companies, because basically what they do, each time we contact a pharmaceutical company or a biotech, they ask us for a portfolio of articles and publications. And the only way to get that is to work with academic institutions. For instance, if you look at the University of Oxford, Pasteur Institute, we have publications with them. They conduct trials with our technology. So it's very interesting for us, because first, we don't have to finance the trials, because usually they initiate the trials, but then we benefit of their results because they help us convince the pharmaceutical companies to use these results and take them forward and potentially transform them into a therapeutic. So to answer your question, the steps are the following. So we start with academic institutions, and then once we have these academic institutions, usually we ask the key opinion leaders at these institutions to help us scout for partners. For instance, last month we had an R&D day at the Imagine Institute, and we had several people from the research institutions I mentioned come over and speak on behalf of Genomic Vision and give their impressions on the technology, and that is of tremendous help for us to then go talk to pharmaceutical companies. So yes, since 2014 we have a team of roughly 10 people traveling the world to convince both academic institutions and private companies to adopt our technology. But the ramp up is we're pretty satisfied with our ramp up, but it takes some time to educate the market. So that's a very good question. So there were several steps. When Aaron decided to leave Institut Pasteur and create the company, there was very quickly a first round of funding with Vesalius Biocapital, which is a VC, a venture capital, European venture capital that specializes in healthcare. And then, for the first 4 years, we were able to live on this first round of funding. And then, then when we signed the deal with Quest, they invested roughly 15 million, so 5 million in an equity investment, and on top of that, they gave us 10 million in the form of R&D milestones upon specific successes in R&D. So for the first years of the collaboration, it was an R&D partnership before it became a commercial license. So we used these R&D milestones to finance the company. But that's a good question, because 10 years is a long time before you become a commercial company. At the time, our cash burn was a little less because we had less employees, but still, the R&D is very expensive and we had to pay all the scientists. So it was a difficult time, but then in 2014, we raised some money in the process of the IPO, and that gave us some runway to finance the transformation into a commercial company. But you're right, the 10 years from 2004 to 2014 were pretty difficult because we needed, at the time, we had almost no sales. The only sales were actually R&D payments made by Quest Diagnostics. That's a very good question as well. So we started to provide our technology as a service only last summer. So we don't have a lot of background information, but it's ramping up pretty quickly because, as you can easily imagine, it's much easier for an academic institution or for a pharma company to adopt the technology through a service because there is no capex involved, no upfront payment, nothing. They pay as they use the technology. So it's gaining more and more traction, and we think that eventually it will actually be the bigger part of our revenues, because we needed to sell the technology as a first step because we need to spread the technology. So as a first step, we focused on selling equipment to then be able to say, these guys are using our technology. But now that this is done and we cover many respected institutions, we are gradually shifting towards a service model.